Okay, hello everyone. So in today's video, I will be talking about unlocking life's code, the power of pluripotent cells. So before we get started, to first understand what pluripotent cells are, they are special cells that can turn into almost any type of cell in the cell body. So this could be skin, muscle, or nerve cells. They can come from embryos or regular adult cells. And the main question here becomes, during cell differentiation, genes that are not required for other lineages are lost or inactivated irreversibly. And so the question arises of when looking at developmental biology and genetics, had there been development that these events did occur, and if so, how did they occur? So with pluripotent stem cells, we're going to focus on two individuals, Gordon and Yamanaka, who did very influential work with pluripotent stem cells. So again, my name is Tala Hamadisa, and I will be talking about the discovery of these stem cells by Yamanaka and Gordon. So pictured here on the left is John Bertrand Gordon, who demonstrated that mature cells can be reprogrammed to an embryonic-like state. On the right is Shinya Yamanaka, who discovered that mature cells can be reprogrammed into iPS cells. So starting with John Bertrand Gordon, before we get started, to look a little bit at the biography of Gordon. Gordon was born October 2nd, 1933 in South England. He then goes on to receive his PhD from Oxford in 1960, focusing on somatic cell nuclear transfer in xenopus. So this was a procedure that was already completed and published by Briggs and King. However, with Briggs and King, they did work on R. pippins instead of what Gordon focused on was the frog Xenopus laevis. And so essentially through his work, Gordon is able to confirm the observations of Briggs and King and basically saying that yes, the later in development that the nuclei is, the less well the cell would be doing. So the main question Gordon wanted to answer is whether the nucleus of a cell that had already embarked on one pathway of differentiation could nevertheless support the development of other unrelated cell types. And so how he did these repeated experiments was by using the normal development of intestinal epithelium cell nuclei and transplanting that to enucleated eggs of the xenopus. Eventually, this would lead to the first cloning of an animal in 1962, but we will get into that later. So continuing this line of work, Gordon does take kind of a break and he accepts a position to do some postdoctoral work at Caltech under Robert Edgar, who worked on bacteriophage genetics. When he returned to Oxford, he then worked as an assistant lecturer in the zoology department and began establishing his research group, where he did these experiments. So after confirming the observations of Briggs and King and continuing more experiments, Gordon eventually showed that the ability of eggs to reprogram somatic cell nuclei back to an embryonic stage. And to better understand how he did this, we need to talk about the methodology that was used. So a UV light from microscopy was effective in killing the egg mitotic chromosomes without harming the egg. A special microforge, that's pictured on the right, was used to break a donor cell without doing damage to its nucleus. So by focusing on an endoderm lineage of embryos, and these were chosen because these cells are very large and had a large content of yolk, Gordon was able to transplant nuclei from larval intestine cells and eventually succeeded in the transplantation of nucleus from a cell that had been committed to a particular type of cell differentiation. Going back and answering that question he originally had of could a cell do this? And this diagram here shows a visual of everything I just mentioned. You have these isolated epithelial cells. The nuclear transfer pipette is then put into this enucleated egg, continues on. You have the partial or complete embryo, depending on which pathway it goes. You have the nuclear transfer. Once you've got into that complete embryo, then these resulting embryos are then metamorphosized into young frogs. The frogs eventually reach sexual maturity, and a lot of these frogs are then able to lay eggs or fertilize eggs. So what are the results of Gordon's work? This was actually one of the first pieces of evidence to suggest that the nucleus of a specialized cell is totipotent, meaning it is able to be changed. So this also caused a little bit of controversy in the topic that permanent gene loss and or modification could not explain loss of total and that this was most likely due to selective and reversible gene modification. Gordon goes on to demonstrate that the nucleus of a frog somatic cell could be reprogrammed to behave like that of a fertilized frog egg. He also mentioned that these somatic cells retained the full retinue of genes and that it was the selective gene expression that accounted for which fate of choice the cell wanted to go. So there were numerous impacts and applications that could be applied from Gordon's work. One of the major ones was that all mammals could now be used for successful somatic cell nuclear transfer. One of the most famous examples of this was the cloning of Dolly the sheep in 1996. And this is depicted in this diagram on the lower left here, where an adult Van Dorset eel was then taken and donor cells from that were cultured in a medium. They were then, through cell fusion and electrical pulses, the nucleus was removed, it was pipetted, the unfertilized egg cell, and then was put back into another sheep. And eventually, this was able to result in the cloned version of the sheep. And this was the first cloned individual. Aside from mammals talking about humans, one of the major advancements of Gordon's work was in therapeutic cloning, which we can see here. There's an enucleated egg type, and using somatic cell nuclear transfer, you can then clone this blastocyte, and with correct pathogenic mutation, this donor cell can then be transplanted back to the donor patient. And this is useful in cases where individuals have diseases, and the somatic cell nuclear transfer can be used to transfer in good cells to help these individuals regain healthy cells. Moving on to Shinya Yamanaka. So before we get into the biography of Shinya Yamanaka, it is important to note that he did follow a lot of Gordon's work and was inspired by the experiments that he did, but he kind of took his own approach on the subject of pluripotent cells. So a little bit about Shinya Yamanaka. He was born on September 4th, 1962 in Osaka, Japan. He originally wanted to become a surgeon and thus completed his PhD in pharmacology at Osaka City University. He then, however, realized that he did not really want to go the medicinal route and wanted to focus more on research. So he then ends up at Gladstone Institute of Cardiovascular Diseases, where he first learned about the NAT1 gene, and eventually this leads him to learning about how to culture ES cells from mice. So moving on and doing work in his lab, he finds an interest in finding how these ES cells maintain their differentiation ability while rapidly proliferating. So the goal of his lab was to generate ES cell-like pluripotent cells from somatic cells without using embryos. And this right here is the most important factor, is that an embryo is not being used. So he followed the work of Gordon and what Gordon found surrounding his process of generating new frogs. And in 2005, succeeds in generating ES-like cells with the 4 factors and named these cells induced pluripotent stem cells, or iPS cells. In 2006, he published a paper in Cell on the successful generation of mouse iPS cells using the 4 factors. The 4 transcription factor genes identified, once combined, were then able to reprogram the fibroblasts and iPS cells similar to that pluripotent state of the ES cells. So to understand this a little bit better, we'll talk about the methodology used. So in step 1, 24 candidate genes are selected and hypothesized that they could possibly induce the somatic cells to become pluripotent. Next, retroviruses were used to insert each of the 24 genes into chromosomes of differentiated mouse embryonic fibroblasts. And by systematically removing these genes, they identified 10 essential genes and focused on 4 that were crucial in generating iPSCs. So primers are then used to replicate this normal embryonic stem cell gene and inject these iPSCs into immunocompromised mice. And this results in teratomas, which differentiated into a 3-germ layer, which is what they were looking for. These cells are then cloned and micro-injected into blastocytes. And here, this diagram here basically shows everything that I just talked about. Looking down specifically at part C, this transcription factor transduction. So there are those 4 transcription factor genes that were notified and showed that they were in iPS cells. So the results, again, these 4 genes, which are highlighted here in red, are identified, and once these 4 combined, we're able to reprogram the fibroblasts into iPS cells that had that pluripotent state that was found in ES cells. And once again, similar to Gordon, this gives us a better understanding of totipotency in cells. And so for impact and application, there are numerous impacts from Yamanaka's work. One of the most noted is the treatment or possible treatment of liver disease. So these induced pluripotent stem cells can be regarded as the optimal cell replacement treatment for acute liver failure. And this is derived from patient-specific cells that can be used as an unlimited source of hepatocytes to treat liver failure. Aside from liver failure, overall treatment of any human disease could be used with Yamanaka's discovery of iPSCs, because we can then study these diseases on a petri dish, which is much more accurate and handy than using animal models. Additionally, this can be used in the future for fertility restoration, but there's not enough research out about that yet. So finally, on October 8th, 2012, Gordon and Yamanaka win the Nobel Prize in Physiology or Medicine for their discovery that these mature cells can be reprogrammed to become pluripotent stem cells. And here in this visual, we can see Gordon on the left and Yamanaka on the right, awaiting their prize. So, significance of their work, once again, they proved that mature cells are not irreversibly committed to their specialized function, and that they do have the ability to be reprogrammed to this pluripotent state, which was a major impact in the scientific world. And so here are all the references that I used. And thank you for watching. I hope this video was insightful and helped you better understand pluripotent stem cells and the 2 scientists that did a lot of work